Welcome students. I am Dr. Arati Pranjale-Ghuse from Bajaj College of Science, Wardha. Today we are discussing the topic chemical nature of gene for M.Sc. second year. Let us first of all focus on early DNA studies. In late 18th century, Johann Friedrich Mischer studied pus, that is contains white blood cells, isolated nucleic material which was called as nuclein, which was later renamed as nucleic acid. From 1928 to 1956, different scientists like Friedrich, Erwin Charagaff, Oswald Avray, Alfred Hershey and Martha Chase, Heinz Frankel Conrad and B. Singer variously defined DNA. Someone defined it as DNA, someone called it as gene. So, in 1953, later on in 1953, James Watson and Francis Crick published paper detailing the DNA structure which was based on published data and unreleased information. And in 1962, they won Nobel Prize along with Maurice Wilkins. Now let us see the chemical nature of gene. The clear concept of gene given by T.H. Morgan, for which he was awarded Nobel Prize in 1933. And the recent work on structure of chromosome and DNA clearly defines the gene as a small segment of polynucleotide chain consisting of hundreds or thousands of nucleotides. These nucleotides are the subunits of a gene. The total complements of genes in an organism is known as its genome. Genes are stored on one or more chromosomes and instruct the cells to make protein. The DNA is found in all prokaryotes and some viruses. And in eukaryotes, it is present inside the nucleus, chloroplast and mitochondria, whereas in prokaryotes, it disperses in cytoplasm. In plants, animals and some viruses, the genetic material is double-stranded DNA, that is dsDNA molecule, except some viruses. In tobacco mosaic virus, that is TMV, influenza virus, poliomyelitis virus and bacteriophages, the genetic material is single-stranded, that is ssRNA molecule. Let us see here some examples of genetic material. DNA, that is double-stranded DNA, dsDNA, found in higher plants, animals, bacteria, animal viruses and bacteriophages. While single-stranded DNA, that is ssDNA, found in bacteriophages and animal viruses. Retrovirus, reovirus, hepatitis B virus, animal virus, these all are the examples of double-stranded RNA, that is dsRNA. While the single-stranded RNA, that is ssRNA, found in animal viruses and bacteriophages and plant viruses. Let us see, these are the building blocks of DNA, which are called as nucleotides. This is structure of nucleotide, which are made up of sugar, phosphate and nitrogen bases. These three are the building blocks of nucleotide. Let us see in detail. Both DNA and RNA are nucleic acids formed by polymerization of nucleotides. Nucleotides are formed of nucleosides and phosphates. This is the chemical structure of nucleotide. You can see here nucleotide contain nucleoside group and phosphate, while the nucleoside contains sugar and nitrogen bases. These are the chemical structure of phosphate, which have 4 phosphorus and having 3 highly negatively charged oxygen. Sugar is the pentose sugar or deoxyribose, which have chemical formula C5H10O5. Let us see one by one all 3 groups. First group is, we will discussing first group, that is sugar group, which is a 5 carbon ring. Deoxyribose is a pentose sugar consisting of 5 carbon atoms 1, 2, 3, 4 and 5. 4 carbon atoms, that is on position 1, 2, 3, 4 of these sugar combine with one oxygen atom and form a ring. The fifth forms CH2 group, which is present outside the ring. 3 hydroxyl groups are attached in position 1, 3 and 5, and the hydrogen atoms combine at position 1, 2, 3 and 4 of carbon atoms. In nucleotides, the pentose sugar is ribose, which is similar to deoxyribose, except that there is an hydroxyl group instead of hydrogen at second carbon atom. The absence of hydroxyl group in DNA makes it chemically more stable than the RNA. Then the phosphate group. In DNA, a phosphate group PO4 is attached to the third carbon of deoxyribose sugar and 5 carbon of another sugar. Therefore, each strand contains 3 end and 5 end arranged in an alternate manner. Strong negative charges of nucleic acid are due to the presence of phosphate group, which we had seen in previous slide. A nucleoid phosphate which contains its bond to 3 and 5 end carbon atoms of pentose sugar, that is called phosphodiester. The last group is the nitrogenous bases, third group, which contains purines and pyrimidines. The entire genetic message that controls the chemistry of every cell of the body acting in a specific way is actually written in the language of 4 nitrogen bases of DNA, that is purines and pyrimidines. The defined sequence of the 4 nitrogenous bases constitute a gene, which may be a few or several hundred base pairs long. Genes are the structure of the blueprints called proteins, which control the infinite variety of life. Let us see the structural detail of the purines and pyrimidines. First of all, purines. Purines are of two types, adenine and guanine. You can see the structure here. This is the basic structure of purine. The purines bases are heterocyclic and 2-ringed bases. They are double ring compounds that consist of 5-membered imidazole ring with nitrogen at 1, 3, 7 and 9 position. You can see here 1, 3, 7 and 9th position. Pyrimidines are thymine and cytosine are 1-ringed bases. They are single ring compounds, the nitrogen being at position 1, 3 and 6, 1 and 3 and in 6-membered benzene ring. A single base is attached to one end carbon atom of pentose sugar by N-glycosidic bond. Then uracil. Uracil is a third pyrimidine, which is little bit different. Adenine, guanine and cytosine are common in both DNA and RNA. Uracil is found only in RNA. These all are the structures of pyrimidines and pyrimidines at a glance. Here we had seen the different structure, all building blocks of the gene, that is, and these all together forms the chemical nature of gene. You can see here the colored different formats of gene. Thank you.